Hello everyone, today's screencast is all about enzymes. This is part of the B1 topic of the biology AQA specification. Let's start, as we always do, please, with the retrieval practice. Pause this screencast now and have a go at these 10 questions. Okay, hopefully you've had a go at them now. So pick yourself up a different coloured pen and mark your answers. So which gas is made during respiration? That is carbon dioxide. The gas made during photosynthesis is oxygen. The movement of molecules from high to low concentration is known as diffusion. Number 4, the rate of diffusion can be increased by increasing the concentration gradient, increasing the temperature. You could talk about the diffusion pathway being shorter, or you could talk about an increased surface area. Number 5, the loss of water is called transpiration. Number 6, the water is lost through the stomata. Number 7, the rate of transpiration can be increased by it being a dry day, so low humidity, high temperature, and when it's very windy. So high temperature, low humidity, and a very windy day. Number 8, which part of the blood carries oxygen? That is the red blood cells, specifically the protein called hemoglobin. Number 9, your heart rate increases during exercise so that more oxygen and glucose are delivered to your cells for respiration, so that your cells release more energy. And number 10, the toxic substance is called lactic acid. Okay, so our objective today is know what an enzyme is, name examples, and to describe their function. So please pause the screencast now and write down the objective. Okay, so what are enzymes? If you get a 6-mark question about enzymes, make sure you get these details down. So first of all, they are made of proteins. So proteins are polymers, but protein are made of amino acids, and the proteins make enzymes. What they actually do is speed up chemical reactions in living things. So I'll give you a couple of examples. Respiration, photosynthesis. Enzymes are essential for those processes to happen effectively and efficiently in the body or in plants, and are described as biological catalysts. So that word catalyst means to speed up, but enzymes are linked specifically to biology, so to plants, to animals. We can use them in industry, but in today's screencast we're going to be talking about how they are useful in digestion. They can build molecules up that you'll see here at the left hand side, but they can also be used to break large molecules down. So please remember that they can build them up or they can build them down. Break them down, sorry. Please make sure you've got an example drawn in your book. So look at this active site, it's labelled on that line. So to get a mark in an exam, if they ask you to draw it, make sure you label that inner line as the active site. This is the enzyme and this is the substrate that it will work on. Okay, the same at this side. We've got an enzyme and 2 substrates this time. So pause this screencast, make sure you've got those key pieces of information written down. Okay, so we're now going to look at digestive enzymes. On the specification for biology, you have to know these specific examples. Now, digestive enzymes are used to convert food into small molecules. So when you eat food, it goes into your stomach, then it'll go to your small intestines where the stuff that we need needs to be absorbed into the blood. Now, when you eat most things, the molecules are too big. So what the enzymes will do is break those molecules down into smaller substances such as sugar, amino acids, fatty acids and glycerol that can be absorbed into your circulatory system and delivered around your body. So if you look here, there's a sort of summary of this at the top. Digestive enzymes convert food into small molecules that can be absorbed into the blood from the small intestines. The smaller molecules can then be used by the body to make larger molecules. So the ones that you need to know are amylase. So amylase is made in the mouth by the salivary glands, the pancreas and the small intestines. And amylase breaks starch into sugars such as glucose. Okay, so the polymer, the large molecule, is starch. The smaller molecule is sugar. Proteases are the next one. This time it's made in the stomach, the pancreas and the small intestines. And the proteases break the proteins down into the monomer, which is called amino acids. The last one are the lipases, so they break down lipids into fatty acids and glycerol, and they are made in the pancreas and the small intestines. Now, please note that the lipases have a little bit of a helping hand. So bile, which is made in your liver and stored in your gallbladder, is actually an alkaline substance, and it neutralises stomach acid. So when it gets into your stomach, it neutralises it. What it also does is it breaks down fat, so the fats that get into your stomach, into smaller droplets. Now, making them smaller increases their surface area, and if you increase the surface area, you actually give more of an area for the lipase to work on. So it helps the lipase because the fat's now got a larger surface area. So the bile helps to break down the fat so that the fat has a larger surface area, so the lipase can then actually break it down into fatty acids and glycerol. So the bile helps the lipase, okay? And this happens in the stomach. So make sure you know these enzymes to do with digestion and you know what bile does. Okay, moving on then. We are now specifically going to look at the activity of an enzyme. So what does the enzyme do? So we have enzymes. There's an enzyme represented by this shape and its substrate. Now, if you look, the substrate's actually got a complementary shape to the enzyme. If a substrate came along that had a triangular shape, it would not fit into the enzyme's active site. So where my cursor is running along now, that is its active site. So if a circle came along or a triangle came along, they would not fit. So we say enzymes are specific. They only work on their substrate, okay? Now, this is the enzyme and substrate. And think about the enzyme as a lock. Think about the substrate as a key. If you tried to take your key for your house out now and get into a different house or into a classroom in school, it wouldn't work because it hasn't got the complementary shape. It hasn't got the correct shape. So that's why we call it the lock. So the enzyme is the lock and the substrate is the key. And this is called the lock and key model or the lock and key theory. When the correct substrate comes along, the enzyme and the substrate join together. The enzyme helps, in this case, to break the substrate up into products. Something that's really key, though, is look at this enzyme. It remains unchanged and it can be used again. So it'll go to another substrate and it'll break that one down. And when it's done that, it'll go to another one and to another. So it can be used over and over again, which makes it really, really good at its job. Okay? So enzyme and substrate, like lock and key, enzyme substrate complex, and then you get your enzyme and products. If you've got a question in an exam, don't be afraid to draw this model. Show that you know this model, show that you know how it works. Okay, so pause the screencast now, make sure you have written this down. Okay, so just before we move on, hinge question. So can you have a go, pause the screencast, write down whether you think A, B, C, D and E are true or false. When you've done that, if any are false, please have a go at correcting them. Okay, so A, enzymes speed up chemical reactions or speed up reactions. That is true. B, enzymes are changed during a reaction. That is false. Enzymes stay as they are. They remain unchanged so that they can be used over and over again. C, starch is an enzyme. That is false. Starch is a polymer that is broken down by an enzyme into monomers, such as sugar. D, enzymes break down large molecules into smaller molecules. That is true, but remember they can also build larger ones up from smaller ones. And E, enzymes are specific. That is true. Enzymes will have a specific active site shape and therefore they will only work with certain substrates. Okay? Okay, moving on. We are now going to look at enzyme activity and temperature. Before I go into the description and explanation of this, please can you draw this diagram into your books and add the labels. Okay, so if you get asked in an exam to do a description, please say what you see. Okay, so there shouldn't be any scientific explanation there. You are just saying what you see. So for this graph, always start with what's on the x-axis. So start with what the temperature is doing. So let's look at the first part of the graph. So as the temperature increases, the rate of the reaction, and in this case the enzyme activity, also increases. Okay, now there's no information about why that's happening yet, that is just a description. Okay, until it gets to 37 degrees, until 37 degrees C, and this is the optimum. Remember, in an exam they like you to include numbers off the graph to show you can read a graph. And then what happens after then? So after 37 degrees, the enzyme activity, in this case we're talking about the rate of reaction, but the enzyme activity starts to decrease, or you could say it starts to fall. Okay, there is absolutely no signs there. Okay, that is just describing what you see. So as the temperature increases, the rate of reaction increases until 37 degrees. This is the optimum. After 37 degrees, the enzyme activity starts to decrease or fall. Okay, so that is your description. Now, the explanation is on the graph. So the reason this is happening in the first part of the graph is because more heat energy causes more collisions between the enzyme and the substrate. So this links to chemistry. If there's more collisions in a certain amount of time, then the rate of the reaction, the enzyme activity, will be higher. Okay, until it reaches the optimum. After the optimum, then the enzymes will denature. Now remember that this is, so denaturing is irreversible. Okay, so please remember that if an enzyme is denatured, it can't be turned back to working again. And it means that the enzyme's active site loses its shape. So it'll change shape, and if it changes shape, the substrate will not be able to fit in anymore. Okay, make sure you've paused that, make sure you've got the description and the explanation, which is all already on the graph. Okay, right, let's have a look at enzyme activity then, and let's have a look at pH. So this time, a description would be that if we, let's do this together then. So if okay, so a description here. As the pH increases, the enzyme activity also increases until the optimum. If you look at all of these, their optimum pH is different. Okay, so pepsin has an optimum pH of 2, which is acidic. Amylase has an optimum pH of about 7, which is neutral, and trypsin has an optimum pH of approximately 9, which is a more alkaline solution. Okay, so we've described the first part of the graph. Okay, so after the optimum, the activity of the enzyme will decrease. Okay, so that's your description. As the pH increases, the enzyme activity also increases until the optimum. After the optimum, the activity of the enzymes will decrease. Now, the explanation is different than the temperature graph because either side of the optimum, it's the same explanation. So this side of the optimum or this side of the optimum, it is about the enzymes denaturing. Okay, so you need to talk about the fact that away from the optimum pH, the enzymes will denature. So away from the optimum, the enzymes will denature. And remember, that means that they will change, the active site will change shape, and remember that that is irreversible. So it doesn't matter if you're talking this side, doesn't matter if you're talking that side, the enzymes are denaturing. Up here is the optimum. Okay, so always look for that optimum on the graph. Okay, right, moving on then. I've written an example of a 6-mark question that I would like you to have a go at. So if you read this question, amylase is required for digestion. Using the lock and key hypothesis, explain the role of amylase. Okay, now please, as soon as you see a question like this, you should be highlighting or you should be underlining the keywords. So I'm just doing this for you now. So amylase is a keyword, digestion is, and lock and key theory, lock and key hypothesis are the 3 key parts that you should be looking at. Okay, so as soon as I saw this question, I would write down what amylase is. Okay, so write down that amylase is an enzyme. Okay, and write down what enzymes are made of. Write down what an enzyme does. Then I would be linking this to digestion, so thinking about what amylase does, so you have to know what amylase breaks down, and then also talking about the lock and key hypothesis. Okay, now remember, for 6-mark questions in science, you have to write in full sentences, but they can be bullet pointed full sentences. So you don't have to put a massive long paragraph, just put some bullet points, but make sure you do complete sentences. Okay, pause this screencast now and have a go. And then when you've finished, unpause and mark your answer. Okay, hopefully you've had a go at that. So look at this mark scheme then. So for just putting that amylase is an enzyme, you would get a mark. So don't forget those simple bits. Just putting the enzymes speed up chemical reactions would get you a mark, such as digestion. Amylase breaks down starch into simple sugars. That would be your next mark. Enzymes are proteins. Again, another mark, just from being able to recall that information. Don't forget the simple stuff. And that they have a specific active site which binds to a specific substrate, and that's your lock and key theory, will get you the next marks. Okay, so see how many marks you've got. Hopefully you have done well on that. Thank you for watching this screencast. Good luck with your revision. Have a lovely day. Take care.